absolute pleasure of mine to giving this online talk. I actually gave 2 sort of physically present talk in Auckland. I mean, quite a few years ago, and it's a beautiful place. Yeah, I used to be at the University of Otago about 9 years ago. So I just realized I don't even put my name in there, but I'm Shinichi Nakagawa, and I'm going to talk about the future of meta-analysis today. So first, I want to start with acknowledgement. This is my group in Sydney, and I want to mention Will Conwell and Collie Callahan, who are the key collaborators of some of the project I'm just gonna mention. So let's jump right in. Okay, citizen science is really changed the way we kind of collect biodiversity data these days, and millions of observations being put in. I think there's a GBIF, the Global Biodiversity Information Facility, that's in Copenhagen, but they have lots of hubs. For example, Australia, we have an Atlas of Living Australia is a hub, and that's a big, database, and the GBIF is a metadatabase, so to say. And yeah, we've been working with Atlas of Living Australia as well. That's, can provide big data. That's one of the foci of this talk, I guess. And you probably, one of those hubs, database hub, is called eBird. And, many of you, I guess, the biologists, computational scientists or computer biologists, they might like going bird watching, and you might actually have an eBird mobile app which you can download. Then you go bird watching with eBird app. What you can put in is this is sort of it's from Alaska. So how many species you have seen? Different species you can put it in. And also how many of each species you have seen. So that's quite important. This is called the A checklist, and that's going to be important a bit later on. And you can just tell like how many people and how long you've done it and how many kilometers, miles you traveled. But this sort of effort amount gets a little bit important later on. So I want you to remember. So that's the eBird. It's one of the database. And using eBird data several years ago, so that's a Collie and Will and myself tried to estimate, all the birds in the world, I guess. And it turns out about 50 billion birds. So that's 7 birds per person. But what's amazing is that was not probably possible, 2015 or 2010, but now that is possible. I mean, we've done the estimate of not just the whole sort of avian population. What we were able to do is each of the species. I think there's about 10,000 species of bird, but 9,700 species of birds, we are able to estimate each species population with very large confidence intervals. Some of the referees are not very happy with large confidence intervals, but lots of guesswork. Okay? What we did here is some workflow is some of the birds populations are really well surveyed, so we can get the actual population number. But most of the population numbers are missing, or species abundance are missing. So we use the eBirds data and also some life history and morphological data birds, because some colorful birds are easy to see. The one species of flocks are easy to see, and the larger birds are easy to see, and also the conservation status might not. So this is the data imputation, right? So we used multiple imputation, and we were able to come up with some guesstimate or estimate of the population size for each bird species. So I just think that's remarkable. We can do this now. But you're kind of thinking, so I thought you came to listen to the talk of meta-analysis. That's not meta-analysis, that's just analysis of big data. So I'm going to tell you how we can use the meta-analysis you probably know meta-analysis is the analysis of analysis because it analyzes the statistics. Right? Each stat provides statistic. We analyze that statistic. That's called meta-analysis. So how can we use citizen science data to do meta-analysis? Before going into the topic, I want to tell you the second law of macroecology. Many of you've heard of the second law of, what is the second law of the thermodynamics, but some of you never heard of the second law of macroecology. That's abundance occupancy relationship. That's an interesting relationship. This is an example from this paper in 2010. This is I think each of the dot is a fish species. And this is a very strong correlation between abundance and occupancy. So what is it? Widely distributed species are more abundant per unit space. Is this intuitive or unintuitive? We don't know, but I mean, that's been used quite often in conservation work and fishing quota. Let's think about the fishing quota because so they say widespread species are more abundant per unit. So it's so wider spread species, we can catch more. Eh? That's the sort of abundance occupancy relationship. So that's an interesting relationship. And it's even more interesting because, there's not many theories in ecology compared to the physics, but Hubble's neutral theory predicts this relationship, abundance occupancy relationship. That's why people take it seriously, even though they don't know the mechanism yet. And here it is, somebody already done the meta-analysis of 279 effect sizes. It turns out to be correlation of this relationship is 0.575, or zeta is the Fisher's transformation of it. But correlation of 0.6 in ecology is kind of crazy number, isn't it? We've done the 80 plus meta-analysis, and I've seen more meta-analysis, but usually correlation is about 0.1 or 0.2. 0.2 is pretty good. 0.3 is almost unheard of. 0.6 is something very special. That's why I think it's called the second law of macroecology, as good as it gets. And this is, funnel plot, and that was a long time ago. It's not quite 20 years ago, but nearly 2 decades ago. But there's a problem for this, yeah? So I said there's about, they don't know the underlying mechanism. They have 10 different theories to explain this relationship. But the first hypothesis is it's completely disregarded in the literature, but it's called sampling bias hypothesis. What it is, widespread species is the easiest to see. Yeah, that kind of makes sense. House sparrows, it's quite widespread, easy to see. Creating the easiest to see, so creating a positive relationship. But if one observe long enough, this relationship would disappear. So let's imagine the sort of 1 kilometer by 1 kilometer space, and you first see the, house sparrow might not, but there's a spot you can see a lot of trees and stuff, and you do sort of really, really thorough search of this 1 by 1 kilometer square, you might not see this relationship, the abundance occupancy relationship. Okay, that's an idea of the sampling bias hypothesis. Also, this meta-analysis, they may suffer from publication bias. People may be just preferentially reporting positive relationship. So maybe, I just think this 0.6 is a crazy number. It's ecology unheard of. So maybe we are, how do I say, overestimating. This is where citizen science data comes in, or eBird data comes in. So I showed you this checklist. We have lots of checklists, and also nearly 8,000 species. We exactly know the distribution of each bird species. Okay, so here's a kind of example. Let's say the example from Melbourne, somewhere close to Melbourne. So in each checklist, we have all the species and how many of each species we've seen. And also from the web page, or you can download, we know the exact sort of distribution of each species, for the nearly 8,000 species. So we can calculate correlation. So this is a beta, yeah? And this might be sort of doing this bird up to filling in. So we can, for each checklist, we were able to calculate correlation. So this is somewhere in North America, this in Europe, and each of the this checklist you can fill in bird watching trip. We can calculate correlation, and we change to zeta because that's good to the statistic on zeta, because I is sort of like, it's bounded by 1 and -1. So I would expect, so precision and this zeta value. So this was the previous meta-analysis, it says about 0.6 something. I would expect this to come down. This, this was probably overestimate. If we use EBA data set, it's so far so good. It makes sense, yeah, hopefully. And here's the result. So result, as you can see, it's a bang on 0. So high, so this is the funnel plot, and I'll take you through. So high sample size. So here's like, the sort of bird watching, like extraordinaire, like, 300, 200 species per bird watching trip. And if you watch that many, it should converge to true effect size. And here is just 5 or 10 species around. And around that time, number, you get lots of sampling errors, yeah? That makes sense. So that's why it's create this funnel shape. And this actually, number of effect size is 17 million checklists. So I would claim this is the biggest meta-analysis ever done, and based on 3 billion observations. Overall effect size is 0. It looks like 0, but 0.015. Surprisingly, this is significant because 70 million, you guys know that there's a relationship between sample size and statistical significance, but I get to that. It's almost meaningless. Yeah, statistical significance there is almost meaningless because of high sample size. What's more surprising is so maybe some of you may not heard of I-square. This is it's an interclass correlation, and it's quantifies the variation you see. I-square 13.5. Most of meta-analysis, this is like 90+%. So what it is what is the proportion of this variation can be attributed to real variation other than sample size. So like opposite side of that is basically 86.5% of the variation you are seeing is purely sample size. And again, this was really surprising to me. Like I've done 80+ meta-analysis, most of meta-analysis 95% plus even this I-square value. So what it is it has to be true 0. So true 0, this relationship doesn't exist. That's why there's no heterogeneity, because if this relationship exists to some population, some area, this I-square value will be very high. It could be 0, but it should be high. But anyway, so black band original meta-analysis, if you read the paper, it says fail-safe number indicates that more than 500,000 unpublished null results would be required to nullify the amazing correlation of 0.6 result, but that's okay because we have a 17 million null results. Okay, so that's cool. And okay, I talked about sampling bias hypothesis. So if you put the effort in, this relationship disappear. And we can see this it's hard to see in a 17 million effect size like this, but the log effort time, 1 log effort time is 3 minutes, 5 log effort time is about 3 hours. And as you can see, it just goes down a little bit. You can't really see. You have to trust me that it goes down. And this significant effect size of overall mean of 0.015 actually disappears if I account for this. But like you wouldn't believe that 0.015 is anything meaningful. But anyway, so I'm just getting, this was a sort of like introduction to the future of research synthesis of meta-analysis. I think future meta-analysis should be a part of data integration. What does it mean? So literature-based one is purely aggregating the study result from published paper is what you know as meta-analysis. This is literature-based. And the citizen science data can be incorporated. And I just showed you some example. Some more might come in later on. I want to give you two sets of examples. So the data integration, easy one to integrate is climate data. So I give you two examples from our lab, and also archived raw data, big data, yeah? I give you two examples on this as well, while talking about the future of meta-analysis. Okay, the first example is this impact rising temperature on the coral disease led by my PhD student. She just handed in, did the exit seminar last week, the SAMI. So SAMI looked at, so coral is, you hear about, I mean, being in Australia, we hear about bleaching, like, danger of bleaching, and it's really tragic, lots of coral, Great Barrier Reefs being bleached. But you may not hear about this. There's another set of dangers. It's a coral disease. And that's a huge problem for the coral as well. And you could imagine as temperature increases, maybe diseases increasing as well. And what we did is we collected 100 plus published work on the coral disease. That's a proportion, and we can just plot like this. And this is coming from all the Atlantic Ocean, Indian Ocean, Pacific Ocean, all across the world. And as you can kind of see, 1990s, I mean, it was like really what's the percentage? I think it was like 3% of coral on average was disease. But if you go to the 2015, that's like becomes about 7%. And we sort of explored it up to 2100, and it's like 70 something percent. It's quite scary stuff. But that's not the part of data integration. That's a pure literature-based meta-analysis. What we did is also you can download it. Many of you might actually download this. Like there's several big weather or temperature database you can download, and we superimpose that data on the meta-analytic data, and we can find as temperature increases, disease prevalence increases too. So that's an example of data integration, not just using the published data. Yeah, because those data were not reported in the original one, but you can download from the web. Okay, second example is amphibian vulnerability to global warming. So continuing the global warming theme. And this was led by the Patrice Potier. He also gave the exit seminar last week. And that's not the main paper. So he needed to first start to get the key effect size, or what we call critical thermal maxima. This is the temperature frogs or amphibians die. To do that, so he collected the heat tolerance or critical thermal maxima for 600 species, 3,000 estimate, because some species are a lot. But here's important. You can see the lots of co-authors, and you can see some of the Asian name, some Portuguese and Brazilian. They are extremely important because you will see, it turns out lots of actually data come from Portugal and China, and they are not published in English. That's only we were able to do this with help of our international collaborators. So importance of non-English literature is critical for the future of meta-analysis. And this been like talked about, not just the meta-analysis, how important it is done by this guy, Tatsuya Amano, a fellow Japanese person. He actually, at the University of Queensland, he's been publishing some really interesting papers. And you might have seen this pulse biology paper. And another one more recently, I think, is a role of non-English language science in informing national diversity assessment. And let's look at one of the figure. Just look at this left-hand side panel. And what it is if you go to, let's say, some familiar country, Japan, yeah, most of like this biodiversity assessment, the differences, Japanese, this yellow and orange, Japanese articles, English articles very rarely referenced. That's same in China, Paraguay, some country like Malaysia, it's almost exclusively English. But this tells you, and many of the non-English literature, or some of them are gray literature, not properly published. This includes lots of important specific, the endemic or native species information. So this shows how important it is to incorporate non-English literature into the synthesis and the meta-analysis. Okay, going back to the Patrice's work. So those are the data of the critical sum maxima. He and his colleague and I contributed a little bit. There was none from Japan, by the way. So I didn't collect any, but I screened all those papers. But as you can see, so the experimental data we got is, as you can expect, lots from Europe and America, but actual diversity is a kind of inverse relationship almost there. There's none in Africa, none in even though we got lots from Portugal, not Portugal, they speak Portuguese, Brazil and other places, but lots are missing. So that is a problem, isn't it? Because, you could actually do the analysis with available data, but what we really want to know, what's going to happen for global warming, what's going to happen to frogs and other amphibians around this really species-rich places. Okay, here comes the data imputation we've been talking about. So we're going to talk about, so we're going to do phylogenetic imputation, which is a very rich information. In this case, because critical, the sum maxima, it has a very strong phylogenetic signal. It actually turns out to be 95%. That's called Pagel's lambda, or phylogenetic heritability equal 95%. So this tree, phylogenetic tree, has a lot of information. So closely related species have a very similar critical sum maxima. So we should be impute this. And also what Patrice did and colleagues did is that we actually did the physical biomodeling. That's like, you can put those aquatic, terrestrial, arboreal in the tree. What's the temperature they're actually experiencing? That, because that's important, because if those body temperature exceeds this critical sum maxima, this is where they die, then they are in trouble. That's an overheating event. Yeah, that leads to, if many of that happens, the species get extinct. So what we found, this was, how many species we imputed? Did it say in here? It's 5,203, and we only had a 10%. But this is the gray bit, is the original data we were able to impute. Of course, with large error for those, where we don't have a very much information, but the simulation shows those imputed values are not biased. It has a large uncertainty, but unbiased. So we should be able to use that. So using that, so that's a critical sum maximum critical temperature actually. But I think the critical sum maximum is another name of it. And the temperature safety margin. So if it would be 0 if the the body operate, the temperature experiencing the frog exceeds the CT max. So those are events. And this is another way of showing it. current temperature, there's already some of the species in terrestrial arboreal species experiencing this, they would die basically, some of the temperature. So that's being threatened. And actually, I'm not putting the pond or aquatic one because aquatic one all survive. So that's very important things to remember. We need to keep our ponds and all those places. Then they'll be even okay when the +4 degree, how do you say, scenario. That's a quite an, but that's, unfortunately, we are tracking to that scenario. No? We thought it was +2 degree scenario. +4 degree scenario, lots of species in, US and, North America, South America, lots in China, Russia, and Australia, and some places in Africa. They're all gonna experience extinction, I think. So that's pretty bad, I think. But anyway, so that's that's a bad news dance. Maybe is that going to be some good news? So I want to talk about the big data and the meta-analysis. 2 examples from our lab, maybe even 3. So it's interesting. So I talk to many computer scientists like yourself, and they say, Shinichi meta-analysis is like, that's going to be obsolete. Okay, is that right? Because, I can kind of understand, because why don't we just, if we have a raw data, why don't we do just a fancy big Bayesian, hierarchical model or something to analyze? Isn't that better? And I agree with that partially, but not completely. And these meta-analysts, Mike Chang and Sedana Jack, they put concept of, that's really interesting, the split analyze, meta-analyze approach. And they think big data is too big, and it's very heterogeneous. You can model one go, but it's more easy to understand. And also, if you are interested in mean and variance, but if you are interested in some sort of like statistics, I don't know, skewness, something different, you can't really model at one go. And if data heterogeneous, like different traits, different location, different year, you can split data and you can do the analysis. And you get the effect size by the results, and you can put together using a meta-analytic technique. And that's probably easier for, rather than huge model nobody understands, we can do the small analysis, get the effect size, do meta-analysis. I think then we can tap into what they talk about. We can tap into meta-analytic toolboxes, and we can do, publication bias, tests, and all sorts of this kind of stuff, which is not really available in the one big data analysis. So that's cool. I think I like this approach because I'm a big fan of meta-analysis, and we want to be a little bit relevant in the future. And I give you some example, and it's again, it's just like it surprises me. Like you can even just download this data. Maybe nobody heard of the International Mouse Phenotypic Consortium. 500 different traits, 100,000 mice. That was like several years ago. It's probably doubled already. 12 institutions around the world. You can just click and download all the data if you like. And using that, we tested this idea of split analyze, meta-analyze approach. So 2 examples. So this is the number of different traits you can get. Some of them behavior, heart stuff, or blood stuff, immune stuff, and it's really interesting. You can just type into International Mouse Phenotypic Consortium, then look at the web page. So one of the first paper, that's not our work, but several years ago, I saw this paper online. They just, that's obvious things to do. You have 500 different traits. What do you want to look at? You want to look at sex differences in the mean traits. But I thought, that's cool. But they show they're different. Okay, yeah, I knew that. But I think the more interesting question is whether those distribution of male, female, the width of distribution, standard deviation, or variance, do they change? So we actually did that using this split analyze, meta-analyze approach, because Meta-analysis, you can do this contrast analysis we use log response ratio or standardized mean difference. You might have heard of Cohen's d, Hedges g. So, but you can actually look at the difference, the sex difference in CV. So we did analyze, meta-analyze, how do I say? Yeah, we split by traits, 200 something traits, and we conducted the meta-analysis on those effect size of the contrast, the male-female difference, yeah? And I want to sort of like, I almost forgot. I want to introduce this paper, tested this difference and width of it's not the, I talked about sex difference in the mean, but the sex difference and width of the distribution is interesting. And this was done by, it's not mice. This is the grades of boys and girls at school up to high school. And what's being said is, as you can see, girls are overall like way more clever than boys. But the interesting thing is, like, girls are all clever, very, very tight distribution. This is a cartoon version. And boys are like this. And this is, it's called the male greater variability hypothesis. And it's been around since the time of Darwin. And also, this was used to explain why STEM subject, this is the boy genius, it's also called boy genius hypothesis. And they look at this and thought like, oh, that's why like lots of guys are in STEM. But we tested this in a STEM. Overall, it looks like this actually. So boys are like terrible in language, especially. But the STEM subject, it's this sort of like girls get variable. So this sort of difference kind of disappears. Girls are still cleverer than boys on average. But that's the comparing the width of not only mean, but the width of the two distribution. And you can get the like, extra insights like this. And there was a male greater variability hypothesis. We can test with mice. And also, I think in medical studies, traditionally, they excluded lots of females subject. And it's quite shocking, like, some area, like 80% of studies are all male mice. And the reason being like, oh, female mice go through this estrous cycles. So we might have to exclude females. And that's the estrous mediated variability. I think kind of interesting, greater male variability assumes there's a lot of difference between males, whereas estrous mediated variability, individual variations kind of similar between sexes, but within individual variations larger. But that's not the focus of the talk. But we wanted to test those two hypotheses about width of distribution using this mice data. And the log response ratio is the mean difference. And this is the difference in the log response ratio, logarithm, what is that? Log of the CB ratio, I think. So, and if you see that this is 95 confidence interval and the mean meta-analytic estimate of morphological traits, I've just highlighted morphology. Males are larger, as you would expect, and there's more variability even on CB, which is corrects for the mean value. Yeah. However, so it's a supporting the male greater variability hypothesis, but life is not that simple. Immunological traits, actually, female mice have a larger, how do I say, values on average, and also more variability, even controlling for mean size. So this supports both hypotheses, and as you can see, some of the traits are sort of in between. So that's kind of interesting. And we use the split analyze, meta-analyze approach using this big data. And another one, this is last example from our lab is the using exactly same data. We kind of thought, oh, we can actually look at the allometric difference between male and females. And it turns out, to my surprise, people don't do this. So what is allometry? Allometry is here's the body size of mice. And this is, let's say this is the size of eye. And this is three different scenarios, yeah? So males and females just differ in a mean, and but the same, exactly same, how do I say, allometric relationship. As body increases, it's usually log scale, and the eye sort of like gets larger for each sex in the same way, but maybe means different here, slopes different. And I'm not going to show the meta-analytic this forest plot, but it turns out, as you expect, for many traits, 60% of traits, slopes are different. Allometric relationship between males and females different. So what is the consequences of this? So there's a nice cartoon done by the Shimek Drobniak. He was a part of our lab till last year doing the Decora fellowship here in Australia. So scenario one is a male and female exactly same size. That's not the case for mice, but assume that, and all the fat or retinal clearance rate, the metabolic rate is all the same, but that's not the case. What people assume is here, so maybe females are small male because, and allometric relationship are exactly the same. So what we can guess without testing female is like, oh, we got the male allometric slope, and all those fat tissue, all that sort of can be scaled. And what you need, because all those metabolic rate and drug stuffs, I guess drug metabolism are closely related. We can assume just you need to reduce the the dosage of a drug. But as we have found out, 60% of traits or many traits related to metabolism are very different allometric relationship in male and females. So if you are giving a two rather than just, for example, you should be giving, just a one here, even though her weight just decreased by 33% or something. So this is the implication. So that's kind of interesting. So this is another example of the, what is it? Split, analyze, meta-analyze, because we split by different traits. We calculated allometric traits for male and female. We look at the difference, and then we meta-analyze those differences. So what is the future of meta-analysis? So I would say future meta-analysis is dim sum. It's not, actually, I think dim sum spells with, what is it? Dim sum, maybe U, I don't know, but the data integrated meta-analysis by split meta-analyze and meta-analyzing. Does it make sense? And, I've shown you some example, but another one I'm working on, last year I was in Okinawa, and it's a really, and OIST, Okinawa Institute of, what is that? Science and Technology. They have like 24 different locations. They're collecting all the noise, acoustic, bioacoustic of 24 locations. And this kind of big data, this is where I think the, dim sum shines, or split analyze, meta-analyze shines, because any statistic we can calculate at 24 locations and we can meta-analyze those. So that's a really nice example, and I'm hoping to work with my OIST colleagues to, make a point about dim sum, how future is a dim sum. And another one is like, is that now a couple of years ago? Yeah, that is the, I wrote this when ChatGPT came out, and I thought I got ChatGPT to screen that, because biggest, like, I guess most repetitive thing we do is extracting effect size or screening a paper for meta-analysis. And I tried whether ChatGPT can do this, and it seems to be able to do it if you put a lot of effort. And I wrote a blog, and actually later on, some of people contacted me and they asked whether we can use it for the, the evidence synthesis or conservation, this kind of AI. And so this is what we call the pyramid of evidence. And here's a meta-analysis, even like it includes the systematic review of reviews. Systematic reviews may not necessarily include meta-analysis, but it usually includes meta-analysis. You probably know what systematic review is, like do the big search, systematically choose the relevant papers, and we synthesize. And those systematic reviews, often with meta-analysis or systematic review of reviews, sometimes with meta-analysis, includes those steps, screening a paper to synthesize the effects. This is a meta-analysis step, and the reporting. And we argued, so it's coming out in the Trends in Ecology and Evolution. We argued like each stage AI can really help. And I've started using this elicit, and it's quite remarkable. So I want to get more scientific. This is what they claim on the webpage, but I'm starting to get actually. So elicit is a software, it's company software. It's not free to do this analysis, but we've used, and amazing. I almost believe this 98% accuracy. It's all like, and they claim they're more accurate than humans. They won't be able to extract effect size, but moderate or some information. This is pretty unbelievable. And we would be able to show, this is mainly, I think, trained on the medical data. So, but we'll be able to tell you how good it is in ecology and evolutionary data soon. So that's also, as Kat said, we are moving to University of Alberta. I'm looking for like 5+ postdocs and PhD student, and that was so one of the, there's a many different projects, including all the synthesis, maybe dim sum project, AI type project, and the meta science project. But the beauty of the thing is you can work on the topic you like to work on within ecology and evolution, I think. So if you're interested, please write to me. And I'm just wrapping up. I think I'm on time. So publication might have shaped our knowledge so far, some work to do. So I've shown so we negated or nullified second law of macroecology. If second law was publication bias, what else could that be? Right? So there's a lot to be still done, I think. And the bright future by combining different types of data, that's what data integration we are talking about, even brighter future with data imputation, because all those big data or database, there's a lot of missing data. And some occasions, if data has a very good covariates, or some traits has a very good phylogenetic signal, which critical sum maxima did, then you should be able to impute. Imputing is just a prediction, right? But prediction is likely to be accurate if you have a good information source. In this case, it was phylogenetic tree. International collaboration, non-English literature, both important. Of course, like AI and those, translation things are getting better and better, but we still know we need to get the native to involved. And this is really good way to reaching out. We have a lots of talk about, global south involving and, EDI stuff, but this is your opportunity to involve those people from global south to collaborate in a synthesis project. Meta-analysis, so meta-analysis, big data, era of big data, we still have a role to play. And theory poor eras coming, and the synthesis sometimes can produce all those new ideas. So I think synthesis or meta-analysis are more important than ever. So as you have said, so future is a dim sum, especially maybe AI assisted. And finally, hopefully, some of you may have done the meta-analysis, but others you might not have. And I think the core of this talk, or I think the message of this talk is everybody should become meta-analyst, or research weavers, or research synthesis. And thank you for listening. Yeah, that's it.